Thank you all for coming. I also want to thank the Peter Wall Institute for Advanced Studies who funded this roundtable. They do this once every 6 months or so. They get international groups together to discuss topical issues. The other roundtables going on right now involve time and life in the universe, and we are our brains, so it's quite a diverse set of topics. And the purpose is to try to forward science or academic pursuits in novel and creative ways. So for this one, we have people from all over the world who've come here to talk about the challenges that ocean acidification presents. If you see someone wearing one of these name tags, they are one of the invitees. Feel free to jump on that person and ask them everything they know about ocean acidification. We also have some posters outside, so afterwards there's a reception with some food and some drinks. And I encourage you to go and check out those posters. Some of the students and postdocs who did the research couldn't be here, so we will be milling around trying to answer questions on those posters if you have any. I also want to thank the museum for letting us do this tonight. They were very kind to us. They were also extremely helpful solving some of the AV challenges. And I really want to thank Megan Vaughn and Nora Brown, who have been assisting with this for many months now, and it would not have come off without their help. So thank you, both of you. For those of you who haven't heard much about ocean acidification, it's been called global warming's evil twin, and many think that its impacts in the ocean are going to be at least as important as other major perturbations like global warming. However, the ecological community only really figured out that this was going to be a problem about 8, maybe 10 years ago. And so we've been playing catch up, trying to discover what all the implications are. And we've made a lot of progress in that time, but we wanted to step back and sort of see what we have now and where we need to get. So I'm going to introduce ocean acidification as a phenomenon and then talk a little bit about why it's relevant here in British Columbia. So what we are doing as a society is producing an awful lot of carbon dioxide that goes into the atmosphere. And when that happens, it then is absorbed by the oceans, where a very simple chemical reaction occurs. The carbon dioxide plus water gives you HCO3 and a little hydrogen ion. That is what is causing the pH in the ocean to go down. pH is a measure of the concentration of hydrogen ions. But it's not just that that's happening. That little hydrogen ion gets a little greedy and grabs onto carbonate ions to make more HCO3. And carbonate is really, really critical in marine biology because it is the building block of calcium carbonate. So if you're a coral and you want to build your skeleton, you need carbonate ions. And if those are getting scavenged by all these extra protons, it becomes much more difficult to build skeletons. The change in pH is also a big physiological stress, and it can impair everything from developmental rates to fish's ability to smell their natural enemies. Well, how fast are things changing? This is the famous Keeling curve from Hawaii of carbon dioxide in the atmosphere, and you can see that we've just been going up and up and up since the 60s when these measurements started. Pre-industrial levels were down here at about 280, and we just crossed, this one is a little bit older, we just crossed 400 parts per million CO2 in the atmosphere for the first time in 2013. Corresponding to that increase in the air, we have increasing CO2 in the water, and that is driving this decrease in pH. So we're already getting measurable changes in the ocean in the pH of the seawater. The IPCC, the Intergovernmental Panel on Climate Change, has just released its report for 2013, and they have a couple of projections of how bad it's going to get. So we've already seen this amount of change in the pH in the ocean. It doesn't look like very much, but it actually represents a 30% increase in the acidity of the water because pH is on a logarithmic scale. So our options are keeping ourselves at an almost impossibly low level of CO2. We're actually going to hit that in the next probably 5 or 6 years, the rate we're going. Or do nothing, and we end up on this trajectory, and that level of pH change would be quite devastating for some species and very interesting for certain ecosystems. This is a global scale phenomenon. This again is that friendly scenario. You see that everything is a decrease in pH, becoming more acidic. This is what I think is probably a little bit more realistic, but perhaps a severe version of it. And so you get these big pH decreases, which are going to affect the rates at which things grow, and on and on. Now in Vancouver, we're already seeing a lot of this. Jeff Marliev and colleagues at the Vancouver Aquarium have been monitoring the pH of their intake water, and they hadn't been paying much attention to that data set until they started having trouble getting their wolf eels to hatch and a few other things that used to be easy. And then someone said, well, why don't you check your pH? And they noticed that it was really low. And then Jeff went back and reassembled this data set, and it was fairly high for quite a long time, and then it starts to become more variable. So this is the sort of the highest and lowest of these two bars, and this is the rough average. And you can see the pH has been coming down here in Vancouver. A lot of that is due to the anthropogenic CO2 that we're putting into the environment. Some of it is also due to changes in patterns of ocean circulation. Now there's lots of species around here that are going to be vulnerable to this. There's a poster out there, I think, on the abalone, which is endangered in British Columbia, and we know that their larvae are very sensitive to changes in pH. Sea urchins are sensitive, these very colorful whelks, that's taken over in West Vancouver. Sea stars actually seem to like it. They grow a little bit faster when you drop the pH. Kelps are impacted both because the pH change messes with them a little bit physiologically, but also because carbon dioxide is a resource, so a lot of seaweeds grow a little bit better. And then you have commercially really important species like oysters. This shot is actually from Washington State, but the oyster growers in British Columbia are also starting to suffer failures in their oyster set, and they are deeply, deeply concerned and starting to invest a lot of research money into solving this issue. The last species that will be affected on here, of course, is us, not just the oyster growers whose livelihoods depend on it, but all coastal people who get protein from the ocean or income from the ocean. But the perspective that I like to take is not just one species at a time when possible, because all those species exist in an ecosystem and they rub shoulders with each other, they eat each other, they overgrow one another, and all of those interactions between species can be quite important. So it's useful to think of all these species and how they're assembled in a food web. So I'm going to cartoon the Northeast Pacific coastal food web here, where you have seaweeds forming the base of the food chain, and then those are eaten by a bunch of grazers like sea urchins and abalone and turban snails. And you also have phytoplankton feeding the filter feeders, things like mussels and oysters. And then some of these things are then consumed by these sea stars. Now we've done individual studies on all of the species up here on the board, and the general consensus is if you're one of these primary producers, so a plant, you're okay. Either there's not much of a change or maybe you do a little bit better. If you are one of these herbivores or filter feeders, notice how they all have calcium carbonate shells. That is bad news for them. So the mussels don't grow as well. They don't recruit into Nora Brown's experiments very well. The turban snails grow more slowly. Kat Anderson's done some nice work on that. The abalone larvae development is impaired. The sea urchins have problems with fertilization and larval development and adult growth. So this whole trophic level is experiencing some, or will experience some problems at carbon dioxide levels that we're expecting to get to in the next few decades. Sea stars, I mentioned before, actually do a little bit better. So at the highest trophic level here, they're fine. They like it. They eat a little bit more. They grow a little bit faster. So if we just look at some of the subcomponents of this food web, like the sea stars eating mussels, that mussel bed habitat is wonderful habitat. There's hundreds of species that live there. But because the mussels are having trouble growing, the sea stars are getting hungrier and growing faster, it's going to be a biodiversity catastrophe, potentially, in mussel beds, because the mussel beds should be becoming reduced in extent, and all the species that depend on them for habitat will have no other place to go. On the other hand, if you look at this pair, the sea urchins to the kelp forests, it works in the opposite direction. So sea urchins do a really good job at eating kelp, and you can see the kelp has to live high enough up here that the sea urchins aren't willing to crawl up to it. With ocean acidification, we think that this species is not going to do well. The kelps, if anything, are going to do a little bit better due to the direct effects, but if the urchins go away, the kelp will be extremely happy about that, and kelp forests may expand. Like mussel beds, there's hundreds of species that live in kelp forests, and so this could actually be good for biodiversity in this particular habitat. Now, I'll caution you that we are not 100% sure that that's correct. One of the best studied kelps in the world is the giant kelp, and this is just its life cycle. Kelps are really, really weird because they go through all these stages. And this just shows you where the effects of ocean acidification have been studied at all, and it's only in a tiny fraction of the total life history. So we do have some work to do to fill in some of these gaps. And we also need to be aware of ecological surprises. So some of you probably know that there's a big sea star die-off going on in Howe Sound and Burrard Inlet right now. I don't think that that's related to acidification, but it does exemplify the kind of surprise you can get when you don't fully understand how the system works. So by way of transitioning to our next three speakers, I want to emphasize that the people that have been here participating in this roundtable really are at the cutting edge of this field, and they've been doing a lot of work to both sort of fill in some of those gaps, extend our knowledge, and reduce the probability that we'll get surprised by these unexpected changes in ecosystems. So our next speaker is Christy Croker, who's going to talk about some of the creative ways that researchers are gathering this data, and then some of the results that she has compiled and collected herself. Then Katerina Fabricius will talk about using volcanic vents as a natural analog, so you can actually sort of snorkel along a pH gradient if you're in the right part of the world. And then Steve Whitcomb will conclude with some advice on what we can actually do about it. Even if you're not a marine biologist or an oyster grower, there are options available to us. So if there's a quick question now, I can field it, but I think it would probably be best to wait till the end. All four of us will be up here to field questions then. So thank you.
So I'm Christy Croker. I'm a postdoctoral scholar at University of California, Davis. And I first want to say thank you to Chris and the organizers for having me here to speak. And then also to all of my colleagues that have been part of this roundtable who have offered a lot of slides and cool videos that you're going to get to see about how people study ocean acidification research. So as a community ecologist, I'm primarily interested in how we go from what happens at an individual scale for an individual organism to the patterns that we can actually see in nature. And this is particularly important in light of environmental changes like ocean acidification that fundamentally affects these individual species by things like calcification or behavioral effects. But the combined impacts on hundreds of species that are interacting with one another can cause really profound shifts in the ecosystem. And I think this is hard to do. Most of our research is still happening on the left-hand side of the screen, looking at how does ocean acidification affect individual species. But I'd argue that it's these emergent effects on ecosystems that are often the most important for people, because this is the scale at which we manage. So I'm going to talk today a little bit about the individual effects, but primarily I'm going to start here with how are we trying to approach these emergent effects on ecosystems. Maybe that will work. Okay, so most ocean acidification experiments start at these single species, and they involve enriching seawater with carbon dioxide and looking how species respond. So this is a setup at Bodega Marine Lab, down where I work at the University of California, Davis. And in each of these jars will be seawater that's enriched with carbon dioxide to a certain pH level, and then we can look at how, in this system, how larvae are responding to ocean acidification. And we've gotten a lot more nuanced in the questions that we can ask. So in this system, for example, we can look at some of these early life history stages that we're more concerned about with ocean acidification. Now we can build that up and move out into the water where we start to increase the realism associated with these experiments. So these are mesocosms that are in the North Atlantic polar regions, where they've actually more or less taken those jars that I just showed you on the previous slide. And now they put them out and they're floating in the water. They're full of water and plankton communities. So we can look at how multi-species assemblages are reacting to the same enrichment of carbon dioxide as they're interacting with each other, and they have natural light and temperature fluctuations. So we get a little bit more realistic here. We can also do experiments. So this is called a FOS experiment. It's based off of the free air carbon dioxide enrichment experiments. These are free ocean carbon dioxide enrichment experiments. And this is pretty dark. This is in Monterey Bay. And that chamber that you're seeing is, again, we're trying to increase the realism so we can try to understand these emergent effects. More or less what's happening is it's open on the bottom so that you can have interactions between the sediment and the seawater. As the water's flowing into the tank, essentially, it's being acidified before it moves in there. So you see now more natural fluctuations in the carbonate chemistry, in the temperature, in the light regime that these organisms are experiencing. Now this one's really cool. So what you're seeing coming at you right now, that's marine snow. So this is deep sea, thousands of meters deep. And this is run by the Monterey Bay Aquarium Research Institute. So they've got this chamber, so this is pretty fun. They are sucking up these deep sea urchins, and then you'll see they move them into this system that they have in the deep sea where they can acidify, again, a single jar that they're moving these sea urchins into, measure the metabolism or even the sometimes growth rates or things of these organisms in their natural environment. I'll let you guys see that guy fall before I move on. So to increase the realism even further, we can use natural gradients. So this is some work that's again done, it's a collaboration across many institutions along the west coast of the United States, including Oregon State University, Bodega Marine Lab, which is University of California, Davis, Stanford, University of California, Santa Cruz, and University of California, Santa Barbara. And so this map here, this figure is actually showing sea surface temperature along the west coast of the United States. And in the areas where we have cooler water, those are often associated with upwelling, where we have naturally high CO2 associated with those waters. So by actually measuring the carbonate chemistry, we get natural gradients in where the ocean is more acidic, and then we can measure what those organisms, how those organisms are responding to those naturally low pH or high CO2 environments. And then last, this is close to my heart, I've done a lot of work at this site, are these volcanic carbon dioxide vents. And Katharina is going to talk a little bit about this in the next talk as well, so I won't focus much here. But these are we have very few examples of these naturally acidified ecosystems where volcanic vents are bubbling carbon dioxide directly into the seawater. And then we can use these as an analog to essentially look into a future ocean. What will it look like at those carbon dioxide concentrations? So what are we finding in all of these experiments? Our general hypotheses are that, one, the organisms will have a harder time making shells or skeleton out of calcium carbonate, as Chris aptly pointed out. The second thing is that organisms that actually use carbon dioxide as an energy source, so any of those marine algae or seaweeds, may actually grow better, or photosynthesis may increase with acidification. And then last, all organisms require energy to regulate their acid-base balance, so maintaining this acid-base balance in more acidic conditions will likely have effects on metabolism. Those organisms that can control their metabolism or their acid-base balance might have higher energetic demands in those same conditions. So I'm going to spend the last half of this talk showing you some general results, which is based on a meta-analysis, which is just a way of summarizing the literature or summarizing all of those experiments that people are doing to try and get at species vulnerability. To answer this question, can we predict which species are most at risk? So this was a figure from a few years ago that shows time on the x-axis and the number of papers on ocean acidification on the y-axis. And as Chris said, we just became aware of this fairly recently. So we've been playing catch up and there's been a lot of experiments trying to understand what does this mean for marine life. And we've got the mesocosm experiments, laboratory, field measurements, and then the total number of experiments. And you can see that exponential rise over time. So one way to approach these emergent or ecosystem effects is to try and get some general sense of what does this mean for a really wide variety of marine species. So a meta-analysis is just a quantitative way of actually summarizing the primary literature. So I take Chris's experiment and Katharina's experiment and Steve's experiment, and then I used statistics to combine them all together and try and see what's the average effect. So the results I'm going to show you are from about 588 unique experiments. And they just measured how species were responding to the projections for the year 2100, which is about a 0.4 unit decrease in pH. And I'm just going to walk you through how you're going to interpret this. You're going to see a bunch of graphs that are very similar. So I've got a mean effect size on the y-axis, which is just the summary statistic. But what I want you to know is that you've got this dotted 0 here, this dotted line at 0. If the data point is below that, it means a reduction in the performance. So for this case, it would be survival. So generally, we see a reduction in survival. If it's above that 0, that means it's an increase in that parameter. Parameter is the best way to say it. The 95% confidence interval just tells you a little bit about how certain are we that that's how species are going to respond to it. And then I have a little number down here that tells you how many studies have actually been done or went into that summary statistic. So here are the general patterns that we actually see in vulnerability. In the way that I've organized them, I've got the most heavily calcified species or taxa over here on the left, and then moving into those species with little to no calcification, like the fleshy algae. And the first thing that I want you to see is that generally, the calcified taxa do appear to be more vulnerable. And this is true across a lot of really diverse types of organisms, from echinoderms like sea urchins to corals. Some of the less calcified or really mobile organisms, fish, we don't see big reductions in growth. Diatoms, we actually see increasing in abundance or growth. And some of those fleshy seaweeds, like we actually hypothesize, do appear to grow better in acidified conditions. Now, this is talking about growth responses to acidification, but obviously there's going to be some unexpected consequences that don't show up in growth. So, for example, we're learning a lot about fish species and that they can actually have these really unexpected responses in their ability to smell their prey or their areas that they want to be in. So this doesn't represent all vulnerability, but it does emphasize the point that calcified species do appear to be some of the most vulnerable. So then we can look at other patterns. So most marine organisms have different stages in their life cycle. This is the larval stage, so this is what essentially marine animal babies. So you've got sea urchins, sea stars, and oysters here. And there's a lot of hypotheses that these early life history stages may actually be the most vulnerable to ocean acidification. So we can test that, again, with meta-analysis. So here I'm showing you larvae, juveniles, and adults, and again, the same axis. So if we think that these early life history stages may be more susceptible or vulnerable to acidification, you might expect to see a pattern like this. And indeed, when we look at the results, we do see for some groups, so this is for mollusks and their survival, that there is this really high sensitivity among the early life history stage, so the larval stage of these organisms. And this is playing out on the coast of Washington and British Columbia, where we are seeing these failure of the hatcheries due to these early life history stages. So now the job is to move from these single species responses and these species vulnerabilities to these larger emergent effects. What does it actually mean in this ecosystem when they're interacting with one another? And that is partially where Katarina is going to take over on this talk. But the general conclusions from all of these 588 experiments that we're able to look at is that there's really negative impacts across a really diverse range of taxa, and calcifiers do stand out as some of the most susceptible. And we're likely to see some cascading impacts on entire ecosystems that may not necessarily be due to these direct effects on growth, but the indirect effects among species. Thanks.
What I would like to talk to you about is these, the phenomena that carbon dioxide in some places doesn't need to be run through bottles into tanks in laboratories, which is very tedious and quite often requires a lot of hours for graduate students to fine-tune things. But there are not very many places in the world where we've got these carbon dioxide seeps. The early studies were in Italy, and there's actually quite a few places in the Mediterranean where CO2 is bubbling out of the seafloor. And then I discovered some in Papua New Guinea when I was doing swim surveys of soft corals and sea fans and just saw those bubbles coming out and said, that's interesting. And now people discovered also some in Japan and in Mexico. But it's really very limited still. We're looking for sites that don't have sulfur, don't have arsenic, are not hot, but are pure carbon dioxide. So basically, there are a place where organisms have had a lifetime of exposure to high CO2, which we normally can't do in laboratories because many of these marine organisms are actually quite long-lived. They may be 5 or 10 or 50 years old. Some corals can be hundreds of years old. So here we've got a system where the larvae are settling in a place that already has high CO2. They're growing up there. And one of the big question is whether organisms can actually acclimatize to high CO2. Like when we go up in the high mountains, our heart goes faster because we're not used to thin oxygen. Similarly, if they have been sitting in, so we take 4 or 5 days to get used to the thin air. But these guys have been sitting in high CO2 for all their lives. And the ecosystems had all the time to adjust and change in order to deal with the high CO2. So we're dealing them using them a little bit like time machines to predict what may be happening in the future in a high CO2 world. Now the place I'm working is a lot of fun. It's in Papua New Guinea, a really interesting, fascinating country, but very remote and logistically quite challenging to work. We do have 3 of these carbon dioxide seeps, and the results I'm showing you are only those that are consistently observed across all 3 seep environments. So we've got some replication to be certain that the differences we've seen between control sites and high CO2 sites are actually just due to the high CO2. But the coral reefs in Papua New Guinea are very, very healthy. They've got very few disturbances. Human pressure is very light. People just fish a little bit with dugout canoes. You see a very healthy, structurally complex and diverse reef environment, full of fish, full of little hiding spaces for crabs and shrimps and invertebrates to live in amongst. And that's only 200 meters away from the seep. When you go closer to the seeps where these CO2 bubbles emerge, even a layperson can see that the coral communities are greatly simplified. These branching corals are very often missing or underrepresented, and these massive corals are taking their space. So overall coral cover is actually not much reduced, but you're getting different types of corals. You still get a lot of fish, you get a lot of algae, you get some oysters as well. So it's a whole community, but it's in a high CO2 community, as we may see in 100 years' time. And in places where the CO2 is touching the coral tissue, lots of algae here, you can see that gas is not toxic to them, but it does, so it doesn't discolor or poison the corals. It's pure CO2, but it just changes the whole balance in the environment. Now in some places this CO2 gas is hitting the coral from underneath and eating out the skeleton. That's an artifact of the seeps because bubbles are coming from underneath. But it also shows that when a skeleton does not protect the coral skeleton, it dissolves. And in more extreme pH environments like here, reef development completely ceases to exist, but some weird sponges can still make a living in there. And seagrass is really, really happy and healthy. Just looks like a golf lawn, really green, thick and lush. So it's a whole heap of ecosystems, sediments, plankton, seagrass and coral reefs that we can study at these 3 sites in Papua New Guinea, and Christy and Jason and a few other people here in the room have studied in Italy. So just again in sort of stills, just to capture it, what you're seeing here, very healthy, structurally complex community, branching corals dominating the coral reef communities. And that is really important habitat for all these reef-associated organisms. We're estimating there's something between 600,000 and 8 million species associated with coral reefs. And many of those depend on the structural complexity of those corals. So it's a huge loss of biodiversity if these reefs are getting lost. At a pH from a little bit above 8 to 7.9, you still have some branching corals left, but not very many. You see the green tinge coming into the ecosystem, that's some additional algae, and more massive corals dominating the community. Now that's something we predict for in a few decades to occur if ocean acidification continues as it is today. Even more severe reduction, end of the century, pH about 7.85. These massive corals are monopolizing the whole space. Now, again, there's still coral cover, but there's not a lot of hiding space for organisms like shrimps and so to live in amongst them. So the habitat is fundamentally altered, but we still, if that is a good proxy for the future, we still will have coral reefs. They're just very, very different and not as colorful and diverse as we're used to see them. Once it gets below 7.8, that's definitely the limit for reef development. Coral reefs cease to exist. There's still individual coral colonies, but it basically turns from coral reef communities into sedimentary communities. And even more severe, some sponges also and some biofilms can live, but it's not much left. Or in shallow water, you get these incredible seagrass communities. Now, obviously, and to summarize ecosystems, it's quite complicated. What we're doing is, this is a map of one of the seep sites. On this side, you see the shore. On this side is the open ocean. So we're doing transects along those pH gradients and basically take a lot of water samples and use loggers to characterize the seawater chemistry and then take measurements along those gradients. We avoid for coral reefs the red bits, which are pH below 7.8 because there's no coral reefs left any longer. That's where the seagrasses are. But we're working always in areas which are pH above 7.8 to 8.1 or so. So basically the changes are gradual and we can use it, like I said before, like a time machine to see what changes are to be expected at what conditions. But averaged overall, and these are the same as a similar level of presentation to Christie's, these are response ratios. If these error bars are crossing the 1.0 line, the effects are not significantly different from between seeps and controls. If they're on this side, this is a 2-fold increase, 5-fold increase, 10-fold increase. And here again, it's on a log scale. This guy here would have 20% of the abundance at the seep site compared to at the control sites. So what you're seeing here is that the top bar here, hard coral cover, is not fundamentally changed at these high 7.8 pH conditions. So the error bars are crossing the 1.0. And there are a few winners in the system. The massive varieties, as I've showed to you, are really doing very well at high CO2. Non-calcareous macroalgae, these flashy seaweed, are doing really well. They're more than double in abundance. And seagrass is more than 5-fold higher than at the control sites. So there's some winners and some losers. But unfortunately, there are many more losers than winners. Hard coral richness, the number of species we're finding per transect, crustose coralline algae that are important signaling mechanisms for larvae to settle. They're basically telling larvae where to settle and establish a new home. They're highly sensitive to CO2. Soft coral sponges all don't like it. And anything to do with coral recruitment. Because Christy has already said, juvenile stages are quite often very sensitive to high CO2. That applies to the corals as well. We've reduced richness of coral juveniles, of soft corals as well, and the density is low as well. So I'm not showing many results. This is sort of a summary of the first visits we've had. There's many more studies that show how individual organisms are changing. But what we're also doing now is compare our results from the PNG seeps, the tropical seep here, with the systems in temperate zones, and with what people are doing in laboratory experiments. And in this case, the bars are control and future CO2 levels of macroalgae in vents and in field mesocosms and in the laboratory. And in all 6 systems, we are seeing a significant increase in algae due to high CO2. Now that tells us that algae are really benefiting from the CO2. They're CO2 limited and they can take it up and convert it into faster growth and or higher biomass, which is important to know if you're an herbivorous animal. On the other hand, if you're a little foraminifera, which are single chips, unicellular organisms but up to 5 millimeters in size, as a unicellular organism, they obviously only have a cell membrane to protect their little calcium carbonate chippies. And we find very steep declines in foraminifera. And indeed, at 7.8, where we still have coral reefs, we don't have any foraminifera any longer. And that is in agreement with the geological record as well. We had 5 mass extinction events in the past, and 4 of the 5 are attributable to high CO2. And in all of those, the foraminifera across the board in most species have severely suffered and disappeared from the geological record. And what we're seeing at the PNG seeps is, and in the Mediterranean seeps as well, a highly sensitive group of organisms here, which are very important sand organisms. Like on the Great Barrier Reef, 30% of the sand in some places is made by these little chippies. Even if this pH is slightly reduced, the tests are already starting to get pitted and eroded, so that's an indication they're really physiologically very, very sensitive to high CO2. I don't have any other results from the seeps, but what I wanted to point out here is ocean acidification obviously can't be seen in isolation. pH of the ocean is sinking as the temperatures are rising. And on the Great Barrier Reef alone, which is on the east coast of Australia where I do most of my work, the temperature zones are moving by 200 kilometers every decade. So if you're coral at 14 degrees latitude, you've got to really hurry up to go down to a temperature that you acclimatize to keep track with the warming oceans, with the temperature gradient, because that goes over 12 degrees latitude. Basically, what the temperature that has occurred at the northern Great Barrier Reef is now occurring in the middle of the Great Barrier Reef due to the speed of climate change we are experiencing. And you probably all have heard about that coral reefs are in strife. Now, the Great Barrier Reef is considered the best managed coral reef in the world. We've got low population density. Australia is a quite affluent country, and we put a lot of money into protecting the reefs. Nevertheless, coral cover, which is on this y-axis here, has declined by 50% in the last 27 years. Now, these are consistent monitoring data from 214 coral reefs up and down the Great Barrier Reef. And on average, we started off in 1985 with 28% coral cover. It's now down to 13.8%, and that's the best coral reef system we've got. We don't have any other data from many places in Indonesia and in Papua New Guinea. So coral reefs are severely affected by things like climate change related factors. Storms and bleaching are severe factors in the whole thing. And also drought-breaking floods are fueling blooms of coral predators called crown-of-thorns starfish, and they all are the main reason why coral cover is going down so badly. Now, it's quite likely the coral cover was about 50% or so in the 1950s before monitoring started. So we may be down to 25% of what it actually used to be. And what these graphs don't even include is the fact that corals are now calcifying more slowly than they used to do 30 years ago. Data from all around the world show coral calcification is declining. On the Great Barrier Reef, the calcification, even of the most resilient corals, is now 15% slower than it used to do. That means recovery from disturbances is also severely affected. So, and we've got no idea how these interactions between temperature and ocean acidification will be panning out, unfortunately. What I just put this slide in just to remind myself to say these declines are not uniform. The far northern Great Barrier Reef, which is away from human population completely, it's upper 500 kilometers of coastline and has got hardly any people living there, probably 20,000 over 500 kilometers or so. Their coral cover has not changed. So here again you've got the whole GBR, you've got the northern GBR on the top right panel, the central GBR, which started off quite low and has been going down fairly consistently, and the south, which is really 150 kilometers offshore, has been hanging in for a long time but has been really severely depleting for the last 10-15 years. So land use is most intense in this middle area, with red being predominantly sugarcane areas, and land use is intensifying. Here the reef is been severely affected by actually by what's happening in terms of water quality up here, because the water is moving down there and the larvae of the crown-of-thorns starfish are moving down there as well. And only the far north is still very pristine. But these 13% are averages, and tourist operators still will take you to really pretty little places. But if you're a scientist, you get onto research vessels, you get to places that most people don't get to see, and you realize in what difficulty the coral reefs are. And just to finish, ocean acidification is on its own is a place we can study on these PNG seeps, because they haven't seen the warming of in 50 or 100 years' time yet. They just have seen ocean acidification. So nevertheless, we've seen severely declining coral diversity. We will see, if acidification continues like this, severely impaired ecosystem functions in the coral reefs of the world, which has implications obviously for all those people like the people in Papua New Guinea who are living off the reefs. They're feeding themselves, they're feeding their families daily by the protein they're gaining from the coral reefs. And there are 100 million people living directly off coral reefs in the Pacific nations. Their food security is severely impaired. So I'll leave it at that. It's a little bit coral-centric, but Steve will again go back and looking at the global picture of what we can actually do. Thanks very much.
It's my privilege to end it all and wrap it all up and try and, what Chris asked me to do is try and give it some kind of context of what we've been hearing this evening. And I'd like to thank you for giving me the opportunity to come, not only for this talk this evening, but for a fantastic meeting we've had this week. And thank you very much for looking after all our logistics and making sure we got here in one piece and actually did what we were supposed to when we were here. But what I would like to do is, I mean, my background is as a marine ecologist, primarily as an experimental ecologist. So all of the kind of things you saw previously, these cool experiments, these looking at field situations, all the kind of things I really have enjoyed doing over my career. But I'm not going to really talk about those kind of things, because what I wanted to really sort of concentrate for the last 10 minutes of these talks is to give you some kind of insight into actually why it is that for the last sort of 10 years or so I've actually thought that this area of research is something that I want to spend my time doing. I mean, there's loads of cool ecosystems out there to go and work in, amazing, fantastic ecological questions still to be addressed. But what is it about ocean acidification that I think is something that I want to spend my time doing? Why is it worthwhile to me? And often the question we're often sort of confronted with as marine ecologists, particularly working in areas of marine ocean acidification, is this idea, people's concept of why the oceans are important. If you sit on your sofa drinking a cup of tea, you might think, well, I'm never going to go to the bottom of the sea. What interest has it got to me? And yet we've seen all these arguments that the guys in Papua New Guinea are dependent on these ecosystems. But it is difficult to make that connection to things that are happening thousands of miles away. And why should I really care? These marine systems, the oceans are always there. I look out and see them, and that's pretty much. I'm not really that concerned with what's going on in them. But I would argue actually that every one of us, no matter how disconnected from the oceans we think we are, has a real connection to ocean systems and also a real dependence on them as well. So, this is just a schematic to give you an appreciation of what we're starting to call the ecosystem services approach. This idea that the oceans and coastal systems actually do jobs for us without even us asking them to do it, asking them to do them for us. And in fact, do them free of charge. And if we had to pay for a lot of these services, we could end up costing us an awful lot of money. So, for example, the oceans provide the food security, so we get our fish and our shellfish from there. But also, this is where we grow aquaculture organisms. We put these organisms into the oceans, and the oceans nurture them for us, and we get the protein back from that. But we spend time out there enjoying the systems as well. So we have recreation. We have recreational fishing as well. It actually makes our lives that much better. But there's the oceans and the ecosystems they support. They also protect us as well. They protect us from coastal damage, from hurricanes, from storms. We also have other resources as well, such as there's organisms out there that might provide medicines or materials that we've not even started to exploit yet. They also have a very important role in maintaining our climate. So as we've seen, there's the amount of CO2 that's going into the oceans. If that CO2 hadn't been absorbed by the oceans, the whole global warming picture would have been so much worse than it is now. So they're mopping up that CO2. And it's not just CO2 they mop up as well. A lot of our waste, our nutrient waste, goes into the oceans as well, and they deal with that without us asking them to. So there's a whole load of things that are provided by the oceans. And particularly in the UK, this has led to an initiative called the Millennium Ecosystem Assessment Initiative, whereby a group of researchers from many different disciplines have come together to try to start to appreciate the true value of the services provided by coastal systems and oceans. And they've split these goods and services, you can see on the right, into sort of major categories of production, of regulation, and cultural services and supporting services here. So food provision, raw materials, I mean, primarily this is almost certainly the first thing any of you would have thought of when we thought about what is it we get from the oceans, and things like fisheries and aquaculture. And as we've seen, ocean acidification could well have a large impact on what we can actually get from the oceans with respect to these. What we've seen is reductions in growth in many marine organisms, and this we think is linked through into the way in which animals use energy. As Christy was saying, that organisms will have a certain energy budget which they can use. They can either use that to grow, or they can use it to reproduce, or use it to build calcium carbonate shells. But they can use it to actually maintain their internal body systems. Now the thing is that energy isn't infinite. There's a finite level to how much energy these organisms to use. So when ocean acidification comes along and makes it more stressful for an organism, they end up spending more of this energy to fight the internal changes, the chemistry changes that they're being exposed to. So that means they have less energy left over, so they have to make sacrifices, they have to make cutbacks. So in times of economic hardship, metabolic economic hardship, they have to decide whether actually we're not going to calcify our shells as much, or we're not going to grow as much. So if these animals aren't growing as much, then obviously that means less protein available for us. Some of these regulatory services I spoke about, but maybe a little bit more difficult to actually conceptually see how they'll be of benefit to us. But gas and climate regulation, this whole idea of balancing the CO2 system and actually producing other key gases that are likely to be implicit in the Earth's climate. Gases such as DMS, which is a cloud-forming gas which is produced by many marine algae, and actually is thought to provide a feedback, a negative feedback, helping the planet to deal with some of the issues associated with global warming. Disturbance prevention, so fighting against these storms and actually putting up ecosystems that protect coastal cities from the storms and storm surges that might otherwise impact upon them. And the bioremediation of waste. If we had to build sewage treatment works to make up for all the waste management that is provided by the oceans, the coastal zone would be absolutely festooned in these sewage treatment works, and the cost associated to us as humans would be enormous. And then it's the sort of things you maybe don't think of immediately, but the cultural value of the oceans and the seas. You know, this idea of, these are places that we go and we enjoy, and we get some kind of pleasure from that. We get some kind of sense of relaxation and maybe life enhancement from these oceans. And it's very difficult to put a price on that. But if we see degradation of coral reefs, some of the things that we automatically assume as being aesthetically pleasing places to be, we can see how the value of these systems will be reduced. And non-used values, I mean, these again are becoming very difficult to value, but there's things like bequest values, and how much is it worth to you as an individual to know that in decades to come there's a healthy marine ecosystem out there for your children and your children's children to still benefit from. What value do you inherently put to that? I mean, none of us might ever see a polar bear, but when asked, did you still want polar bears to be there? Well, yes, we do. So there's a value to that. So there's values associated with things that we'll never actually use, but we just value them being there. Option use values. This idea that the oceans could actually contain a whole suite of organisms, chemicals, or processes that might be of direct benefit to us in the future. We hear about how the rainforests are a huge depository of medicines that we still could go and tap into. The oceans are similar. We're forever finding new chemicals that could be of direct benefit for us. For example, there's been efforts in the UK to isolate sunscreen chemicals from plankton. Plankton live up in the surface oceans and are constantly exposed to UV radiation. So they develop these sunscreen chemicals within them that we're thinking that maybe we can use and adapt to use for ourselves. So all these option use values that could be valuable in the future. And these supporting services, this idea of nutrient cycling, the oceans actually cycle much of the nutrients on the planet to fuel, linking back into production and productivity. Nearly 80% of the nutrients that fuel primary production in the coastal zone go down through the sediment system and get processed and turned into usable nutrients for primary production. Resilience and resistance, this idea that biodiversity, by having a large suite of organisms out there, gives the system some kind of resistance against change. And also biologically mediated habitats. There's animals out there that change the environment in which they live to support things like biodiversity. So, what have the oceans ever done for us? Okay, well, hopefully now I've given you a little bit of an insight into some of the things. So what have the oceans ever done for us? Well, food, transport, entertainment, energy, oxygen, hygiene, protection, relaxation, the climate, and public health. But apart from that, what have the oceans ever done for us? Now, I think it's important that people begin to see the connection between, just because you're not next to the ocean or you don't see it every day in your lifetime, it doesn't mean that you're not going to be taking a direct benefit from the things that go on in the ocean. And also, therefore, issues such as ocean acidification, which can have such marked impacts on the ability of marine systems to deliver these goods and services, should be something that we're all take an interest in and have a concern about. So what can we actually do to help? Well, the obvious thing is reduce CO2 emissions, and I think that's a given. And perhaps in days of increasing energy costs, that's something that's not just of benefit to the oceans, but it also has a personal benefit as well if we can reduce our energy consumption. But I think we can be fairly realistic that whilst it would be fantastic if we think we could cure something like ocean acidification purely through the reduction of CO2 emissions, I think we're realistic enough to realize that actually the political landscape is such that we're not going to be able to meet some of these really ambitious targets. And therefore, we need to think about other ways in which we're going to be dealing with ocean acidification. And one of these is about managing and protecting the existing ecosystems that we have there. And as a group, one of the most interesting discussions we've had this week is, are there actually things we can do in the coastal system that will in some way future-proof these ecosystems against the change which is inevitably coming? And things like reducing the amount of organic nutrients we put into ecosystems can have a significant effect on how organisms and how ecosystems respond to ocean acidification. So by managing something we can have a control over, puts us in a more favorable position to deal with things that are coming at us from a global scale. Things like maintaining levels of biodiversity, to give us that sort of that pool of species that even if we do lose some due to high CO2, there are others there that are performing the same function and the same service that we need them to perform. Maintaining the structural complexity of ecosystems around us. So if you have a coral reef offshore and you know that ocean acidification will in some way start to degrade that reef, don't go out and dynamite fish it and start to blow it to pieces and allow the acidification to exacerbate the effects. So all these kind of local scale management approaches can be used to mitigate what is in essence a global problem. There's even some technologies or schemes that have been proposed to reduce the impact. And in fact, there's been a lot of effort and and resource put towards are there novel and ingenious ideas that we can come up with which might give us a way in which we can mitigate the impacts. And in fact, one that's been recently suggested and recently received some funding, it's a concept about can we genetically breed corals that are more likely to be resilient to environmental stress. And can we actually design some super corals that might help us and see us maintain coral reefs in a high CO2 future? And then there's some of you may be aware of engineering solutions, geoengineering to change, to start to address some of the issues associated with, primarily these are to do with warming, but also to ocean acidification. Now, in no way as a scientist am I advocating either of these two at the bottom, but I think it's very important that we keep our minds open and we explore fully our human ingenuity and actually look to see whether these potential techniques exist. But what we absolutely must do is ensure that when we look at techniques such as this, we're fully aware of the implications of doing it. We're already running a global-wide experiment with CO2 that's having pretty drastic effects. The last thing we want to do is start running another experiment to exacerbate things by fiddling even more with a system that we don't fully understand. So I think I give you this summary of some of the geoengineering techniques because I think it's really good to be aware of all of these things. And actually, some of them we've seen in the past that whilst initially have seemed like very good ideas, that when we understand the impacts of the full scheme, the downstream effects have been a lot worse than we thought they initially would have been. And one example of that might be familiar to people in the audience is this idea of ion enrichment, creating plankton blooms that would supposedly draw down CO2 from the atmosphere. So whilst that went through a period of being considered a feasible solution to mopping up more CO2 from the atmosphere, it then became clear that when we understood the more wide-scale impacts of that technique, it became clear that actually that was a solution that was going to give us far more problems than the problem that we were trying to cure in the first place. So I think we need to be open-minded about different techniques, but we need to be fully informed and we need to generate the information. So we make decisions based on evidence and not just on a knee-jerk reaction. So hopefully what I've managed to do in the 10 minutes is to convince you that actually, although ocean acidification is taking place out here in the oceans, we all have this connectivity to the oceans, and even though we might not think of it every day, we are benefiting from the things they do from us. And ocean acidification can have considerable impacts on a lot of these goods and services. So it's something we should care about. So if we're back on the sofa drinking our cups of tea, maybe we need to be just a little bit interested in what's going on the sea floor. So I'll leave it at that. Thank you.
We can hear you. So one question I have is, as we see ocean acidification occur, would we possibly see a shift from organisms that are invertebrates, like the ones that are eating the algae, etc., the ones that are producing shells? Would we see a shift from them to, say, another species that doesn't grow shells, like fish, for example, that might take their place? And in Hawaii, the trophic group, the group of animals that actually does most of the grazing on algae are fishes. There are very few shells. So, just curious if that's like something that you discuss or think about as possibly just being a shift from invertebrates really being the dominant grazers to fishes and other organisms.
Sounds like one for you,
Yeah.
actually.
So I've done some work at these volcanic carbon dioxide vents in Italy where our dominant grazers are typically calcified invertebrates, so sea urchins and mollusk snails. And what we do think about this and are interested in it, and what we see is that we lose a lot of those hard-bodied, hard-shelled organisms, and we see these blooms of small crustaceans, so things, I call them the pods, amphipods, isopods, taneids. And these are crustaceans that are also herbivorous. So we do see them compensate somewhat. They actually increase in abundance when some of the hard-shelled organisms are lost. However, these are fundamentally really different species or types of species, so they're much smaller, they have less biomass, and we are concerned about whether or not they can actually graze the same amount as some of these large-bodied shelled organisms. And then what that actually means for higher trophic levels, because now you've replaced a couple really big, heavy biomass, or with a lot of biomass, invertebrates with a lot of these little tiny ones, which are unlikely to be able to feed the higher trophic levels like fish. Now, we have a different instance in the coral reefs, where I'll speak to it quickly, and then we'll move on. Where as you said, the other grazers are fish. So we're a little less concerned about the grazers in that system because it's likely that the fish may be able to actually do the grazing.
Yeah, except that, for example, in the Caribbean, the herbivorous fish have been heavily fished for decades now, and the sea urchins, Diadema, have become the key herbivores. And unfortunately, through a disease, we only found out how important they are because all the sea urchins have now died back in many places, and the macroalgae are now overgrowing the coral, contributing to the decline in coral cover. So in many places, we are just underestimating the role of many of these hard-bodied invertebrates. They're still playing an important role. Most of them are not visible during the day, but come out during the night. And only when you do a night dive, you realize how much of it is actually there, how busy they are scraping the bottom and changing the ecosystem. So yes, CO2 will change the ecosystems, but in many cases, we scientists are still completely unable to predict the ecosystem effects in the bigger picture because it's just so complex and we've only started looking at them. Do you want to add to that?
I can talk loud enough, I think. I noticed that you said that there would be greater plant life if there were higher CO2 concentrations. Of course, in photosynthesis, CO2 is used up in the process. Do you think this would be beneficial in checking the balance?
It will be helpful. There's a big discussion of blue carbon, which is seagrass and other tissues like that. So if we can get seagrass to grow more, they soak up carbon dioxide, they store it in their leaves or shoots, and then maybe that rolls downhill into the deep ocean and gets buried down there. So that will help to mitigate some of the effects of extra carbon dioxide. But I don't think that those coastal habitats can fully keep up with all the extra CO2 that's being added. So we should certainly try to preserve our seagrass beds to the best we can, but we need to look for other things too, or expect carbon dioxide to keep going up.
The other considerations with that as well is, of course, ocean acidification is not happening in isolation. So, whilst increased CO2 might be beneficial to some algal species, maybe rising temperatures will not. So there'll be a trade-off between warming and acidification. And also, any increased productivity from the algae, it might be a case that if we've lost the grazers, then that kind of extra productivity might actually become a problem in the ecosystems. And instead of going through sort of invertebrate and then vertebrate food chains, might suddenly start to fuel the microbial food chains. And if we get increased microbial activity, bacteria and what have you, rotting things down, we might have instances of increased deoxygenation of systems as well. So it's one thing that I think we spend an awful lot of time talking about this week is the interplay between some of these really big climate stresses and how sometimes they act to exacerbate a problem, sometimes they act to sort of cancel each other out. but often the complexities are very difficult to get a handle on.
You didn't mention the possible reduction of the size of fish and other creatures due to the difficulty, the increased energy required to breathe in the high CO2. You'd like to comment on that?
I'm going to slightly dodge your question by talking about sea urchins first. We have done some work on sea urchins. They do grow more slowly, and one of the things that means that is instead of taking 5 years to grow to legal size, it takes 8 years to grow to legal size. And because a few of them will die every year along the way, that means that fewer of them get into the fishery. And even when they're there, they're not growing up to those big granddaddy and grandmommy sizes, so when you're harvesting them, they are smaller. Fish are less sensitive to ocean acidification. Yes, they do have to do a little bit of physiological work to regulate how acidic their blood and body tissues are, but as far as I know, it seems like the bigger effects on fish growth are going to have to do with their food supply and temperature, and ocean acidification influences food supply, but it's a little bit more difficult to predict with fish.
In the Great Barrier Reef, you were mentioning how it's moving down and they'll have to migrate quite quickly in order to keep up. Have you ever been tempted as a researcher to seed them and move them?
The Great Barrier Reef is relatively well connected through larvae, because corals release larvae in the water column and they're rafting down the Great Barrier Reef, in particular from the north to the south. So the seeding to some extent is happening naturally for us. So again, that's one of those ecosystem services nature provides for us for free. But people are talking about the need to actually help them accelerate. For example, corals are associated with tiny little algae in their tissue. So corals, like plants, make photosynthesis. And these algae are particularly temperature tolerant or temperature sensitive. So people have been thinking about taking algae from the north to the south to help the corals dealing a little bit better. But the escape to the south is limited just through geographic boundaries. They can't continue going south further and further because they run out of Great Barrier Reef. And the same exists further south in Australia for kelp forest. They can't go further south because there ain't any continents any longer. So to some extent, yes, ecosystems are shifting. And we're finding now corals in Japan where there've never been any corals before, and so to some extent ranges are expanding, but at the same time ocean acidification is even more unfavorable in the polar regions than in the tropical regions, because warm water doesn't hold as much CO2, so it's more readily available for corals. And then colder waters, we talk about saturation. Basically, warm beer doesn't hold any CO2, so it goes flat, and cold beer does. So in the cold areas, ocean acidification is actually a huge problem, because many of the waters are sort of boundary of getting corrosive even for a few weeks now in summer. So there's only a limited amount of time they can use this escape into latitude to deal with the issue.
So, just a quick follow-up. If I follow you well, you are telling me that the fish will move from the tropics, right, to the poles because of climate change. And then as they go to the poles, it's acidic. So they're moving from frying pan, which is the tropics, to the fire. This is scary, isn't it? Is that true?
Unfortunately, yes. And what is really unfortunate about the whole thing is that what we're doing is irreversible at a time scale of hundreds of thousands of years. The engineering solutions that Steve introduces, unfortunately, none of them work at this stage to combat ocean acidification. We have no means to get the CO2 out of the air back. So what we're doing today in the next decade will affect people in 1,000 years' time. So it is irreversible at a time scale we humans care about.
Follow up to that, and I won't hold the mic because I have to gesticulate. One of the predictions based on physiology is animals have a thermal performance or range from cold to warm. And one of the effects of extra CO2 is that that range shrinks a little bit. So one of the things we've been discussing at the workshop this week is if your range in latitude from warm to cold is like this now, that may shrink as well. So that's one of the wild cards that we don't understand very well yet, but will certainly be important.
I'm a sucker for those scare stories about jellyfish taking over the oceans. Will ocean acidification affect jellyfish in either way?
I think there's been one study on jellyfish, and we have no idea what it means for jellyfish. But there's nothing that's pointing us to it's going to be a boon for jellyfish or anything like that.
But there's nothing that says it isn't either.
Geoengineering, you talked a little bit about that, and you said the iron sulfate was an issue. Is there any, there was an example of that last year here on this coast, where there was a kind of rogue experiment. Is there some examples that you know of that are actually look like they're going to work and work well?
The solution that particularly in Europe, that's attracting the most attention at the moment is the geological sequestration of carbon dioxide. So capturing carbon dioxide at source from power generation sites, and then shipping it and pumping it down into the rocks. So either putting it back into the old oil and gas reservoirs that have become depleted, or putting them into geological structures which are currently full of with brine. So these are called saline aquifers. And there is a huge, it's a huge global effort now to explore the potential for capturing that CO2. In essence, putting the carbon back into the geological reservoirs where we've taken it from. Technically, this approach is durable, and there have been demonstration projects where carbon dioxide has been stored successfully and continuously for many years now. And there's a capacity in the geological formations to store huge amounts of CO2, enormous amounts of CO2. My involvement in that activity is primarily, I want to know whether it's safe. I want to know what the risks are associated with leakage. And early comments from engineers that these systems won't leak, I think were never fully believed. But I think the more I've looked into the potential impacts of leakage, the more convinced I have been that should leakage occur, it will be fairly localized and it will be fairly short term. Now, the difference with putting something like CO2 under the ground compared to what might be perceived to be other dumping activities in the ocean is the fact that it's not a do or don't situation. We are already dumping CO2 in the oceans. We're doing it via the atmosphere. And I think what we need to start to do is to assess the risks in context with the risk of not doing something like this. I don't think geological storage is a cure-all for warming or for acidification, but it may be a tool, so the jury's still out, to buy time and to reduce the amount of CO2. Because every ton of CO2 that isn't in the atmosphere isn't going into the ocean and isn't contributing to ocean acidification.
Just a follow-up. If we assess the risks and say, okay, it's too risky to continue doing what we're doing, do we have the social ingenuity to change what we're doing? That's really the question that's out there. I mean, can we govern ourselves to do something different?
That would be an interesting question for one of the other Peter Wall groups. We Are the Brain, I think. I don't know. Wouldn't it be nice to think we did? But I'm not convinced.
But I'm convinced the technology is almost there to really switch to renewables. And I mean, we are mobilizing a lot of money to go to war here and there. And I think we really needed to deal it with the emergency it has and mobilize resources. And I mean, most people speak about 1 or 2 or 3% of gross national product to be invested into changing completely from fossil fuel to renewable fuel. The technology is there, we can do it. The political will has to follow, and the power structures have to basically adopt this as a new opportunity instead of a threat to existing market structures.
One last question, I think we have time for. Yeah, just to follow up on that, can you say a little bit about the distribution of the effects of ocean acidification on a global scale? For example, in the case of climate change, we hear about drought in Africa causing famines and war and whatnot. Can you say a little bit about the distribution in terms of human population effects on a global scale?
Can we cover? Well, I'll leave you to talk about the development. I'll just talk sort of on a geographical scale of what we know about. I mean, we've already spoken today about the chemistry of ocean acidification is such that, you know, polar waters are at the forefront of the chemistry changes that we're seeing due to the nature of cold waters being more likely to dissolve the CO2. We've also noticed that actually, whilst this was a phenomena that was initially identified in sort of oceanic systems, offshore systems, the more we look at coastal systems, the more we see that actually CO2 impacts and ocean acidification impacts are that much greater in coastal systems because of all sorts of other things that can contribute to acidification. Influences of large rivers, microbial processes, all of which are contributing to that. But it's also incredibly patchy. And over the last few years, there's been a very strong effort to try and understand the spatial patchiness and what drives that. Because when it comes to predicting impacts, it's essential that we actually understand the scale of the problem at different places before we start to extrapolate up to potential impacts. I think you want to perhaps say something. Yeah.
So one other side of that in terms of human vulnerability to ocean acidification involves seafood protein. So almost half of the protein that people get from the ocean is from shellfish or from some of the species that we're most concerned about with acidification. And some of the analyses that are being done show that it is the developing countries that are most reliant on those shellfish products, shellfish protein. So the countries that are emitting the CO2 are not going to be the same countries that are actually, countries and people that are feeling some of the, at least the shellfish protein or the food security issues associated with it.
At the same time, it's probably the Western country who feel the economic impacts of sea level rise the first. I mean, New York and there's so many places that are low-lying and sea level is rising. The predictions are a bit less clear than for ocean acidification and warming, but it's definitely a factor that will cost us trillions and trillions of dollars if you have to lift up runways of our airports and so on. And it will cost a lot of lives in Bangladesh and many places that are low-lying. It also will cost many lives because of the droughts, flood cycles. Australia has got the most severe wildfires right now since it's a long time, very, very early in the dry season and so on. So, I mean, the IPCC report again predicts the climate instabilities will affect developing countries, unfortunately, much more than the developed countries where most population is. But we are feeling it in our hip pocket as well. And I think most economic models say it's definitely much cheaper to invest into renewables today than to fix up the mess we are creating in 20 or 50 years' time.
I think maybe we'll take the discussion into the reception, if that's all right. And I think, so the reception will be out there, and just a reminder not to take food or drink into the collections. And if you want to talk to anyone with a name tag who's part of this workshop, then feel free. That's okay. And anyway, would you guys join me in thanking the panel for really great talks tonight?